So before we get into honeybees, let's just remind ourselves that not only do a number of the things that are transmitted between bee colonies impacting honeybees, but they also impact many of the wild bee species as well. So we have a duty of care as responsible beekeepers to make sure that we're doing everything we can to keep healthy colonies so that we're not spreading diseases into the environment and decimating our wild bee populations. Before we can really talk about what is an unhealthy hive, let's quickly talk about what is a healthy hive. A healthy hive has a good population size, nice densely populated frames. The activity level at the hive entrance is, on a warm day, pretty intensive, but it's gentle, calm, happy behavior. You don't see any battling. You don't see any bees trying to sting each other. Nice healthy brood pattern, so nice full brood pattern, healthy looking brood, and bees that are nice and young, fresh looking. So one thing that we commonly say at Bee Care Institute is that a strong colony in a small hive might be a weak colony in a large hive. So you got to make sure to right size your hive. And prevention is worth a pound of cure, as they say. So prevention, in order to prevent many of these things from impacting your bees, you want strong hives, you want good brood production, good nutrition, young prolific queen, and then good hive volume, so good colony strength. You want to limit your disease transmission, and you do this by how your apiary is set up so that bees are not going from one hive to another, going into the wrong hive when they come home. You want to avoid inducing robbing, so make sure you're not using that Boardman feeder full of sugar water at the front. Make sure if you feed your bees you don't spill. If you're pulling honey, make sure that you're not making a honey mess on the outside of the colony. You want to reduce the amount of an invitation that you're setting out. And you also want to make sure that when the dearth comes, you have either your robbing screens up or you have a smaller entrance with the entrance reducer set to smaller. And then you want to isolate your apiaries as much as you can. Now, in an urban environment, that's not always possible. But if you have an option of keeping your hives a little further away from other colonies, other apiaries, it might be good to make it so that if another beekeeper is not doing what they're supposed to do, your bees aren't going to suffer, or your bees aren't going to then become vectors to continue to proliferate some nasty thing into the environment. And you want to practice good hygiene. So maybe not so much colony to colony within your apiary, but if you're going between apiaries or if you're working at your mentors and then you're going to come back and work on your bees, just make sure that you're wiping your tool down with isopropyl alcohol, patting isopropyl alcohol between your gloves. Some people like to light their smoker and stick their hive tool in their smoker fire for a minute. That's fine. Don't do it too hot and for too long or you'll lose your temper on your tool, but a little bit of heat probably isn't going to do anything too bad to your tool. But you just want to be thoughtful about not taking things from one colony and putting them in another unless there's a specific management requirement for you to do that. You also want to make sure that your hives are situated so that they have good ventilation and they're in an area where there's not like a lot of, a humidity buildup. So you want to make sure they're up off the ground. You want to make sure there's some airflow, not on the lowest part of your whole property where it kind of gets muddy and never really gets dry. You want to put them in a slightly different location if you're running into some of the problems that can arise from damp conditions. So we just looked at prevention as a part of the first part of the cycle. So you want to keep strong colonies and limit disease transmission. Next part of the cycle is that you're going to do monitoring. So you're going to do testing, you're going to do observations, and you're going to keep really good notes. And then if something arises where you feel like you need to take an action for whatever reason, you do the thing. You apply the treatment, you do the activity, whatever it is to resolve the issue. And then you want to come back and you want to evaluate. Whatever I did successful? Did I actually address the issue? And part of this is going to be retesting, rechecking, going back and doing the same kind of thing you did in order to discern that you needed to take an action in the monitoring phase. And then you're going to compare your results from the monitoring phase. It's part of the reason why it's so important to take good records. And then you're just going to keep on doing that over and over and over again. So the culprits we're going to be discussing in this presentation are parasites, so your varroa mites and your tracheal mites, some of the viruses, parasitic mite syndrome, bee paralysis, sac brood, some of your bacterial issues, American foul brood and European foul brood, some of your fungal issues, such as nosema and chalk brood, some conditions like dysentery, chill brood, and colony collapse disorder, and some secondary pests, wax moth, small hive beetle, predators, bears and teenagers, mice. Some of the other threats we're going to look at briefly, or some of the other threats that are out there, are poor nutrition, pesticides, herbicides, fungicides, and clumsy beekeepers. So we're going to be looking at many of these things through an IPM lens. IPM stands for Integrated Pest Management. And this is a way to rethink how we approach diseases. In the instance of the varroa mite, mites are becoming resistant to many of the chemical treatments. Overuse and misuse and near total dependence on chemicals have led to mites developing resistance to the chemicals most frequently used. Miticide misuse, contamination of comb and honey, use of chemicals and antibiotics in the hive have resulted in beeswax that is contaminated with these chemicals. This affects the health and stability of the hive as well as the economic value of the wax. Misuse has resulted in honeybees being contaminated as well. So diseases associated with mites are difficult to treat. No effective economical method for treating viruses associated with parasitic mite syndrome. Mites now are resistant to some of the treatments, Apistan and checkmate, in some areas. So we're thinking about how do we treat mites without using chemicals? And on the bigger scope, how do we treat all of the issues that impact a hive, starting with sort of the least invasive options to the more nuclear, high potency chemical options. But some of the drawbacks to integrated pest management is that it can be more labor intensive, can require a little bit more time or more trips to the apiary. And there's no real simple answer. Each individual beekeeper has to kind of think their way through it for themselves and learn and get experience and continue to develop new practices and keep up to date with the current research. And then if you do something one time, it may not consistently work. So you may have to try something else. Or what might work for you may not work for your neighbor or your mentee. What worked for them may not necessarily work for you. The best thing you can do is make sure that you are doing really good record keeping, that you are following the cycle of prevention, monitoring, action, and remonitoring, evaluation. So integrated pest management is a decision-making process. You're looking at pest-host interactions, considering thresholds, like how much of this issue can I have in my hive and it be still a fairly healthy hive. Multiple tactics, so it's not just one answer to that question all the time. And it must be safe, profitable, and environmentally friendly. So the knowledge of the life cycle and seasonal rhythm of the pests, knowledge of the life cycle of mites, for instance, both varroa and tracheal, also allows you to know what time of the year the mite populations are at their highest level and when they most impact honeybee health. Determine when the pests are most susceptible to treatment. Varroa, for instance, reach peak population in late July, August, and September. As beekeepers, we want our colonies to have low varroa mite populations during the period of time when the winter brood is being reared, because this is the time when the mites are going to be parasitizing on the bees and shortening their lifespan, and they're going to have a harder time making it through winter. So the winter brood is the population that will carry the colony through and begin the colony expansion in the spring. So the bees begin to lay winter brood in late August and continue through October, and perhaps even beyond. Giving your knowledge of host of mite interaction, controlling mite populations during this period of time is essential. Tracheal mites, when present, reach peak populations during the winter months when the bees are confined to the hive. These mites can move from one bee to another with ease because of the closeness of the bees, since they're clustered up. Keeping populations low at this time will enable bees to live through the winter and begin brood rearing again in the spring. And then you have to consider your economic thresholds. So research has indicated that in general, if more than 50 mites per day collect on a sticky board, treatment is indicated. That should say 5 mites a day are collected on it. Treatment is indicated. The economic threshold is that the threshold above which you have economic injury or possible colony loss. Treatment only of colonies or apiaries that need treatment saves beekeepers money and reduces the chance that mites can form resistance to miticide. Using methods other than chemicals assure that better bee health and reduces the possibility of contaminating honey and crops, and also reduces the chances of developing resistance to miticides. So the IPM rules. Again, you're going to detect or monitor for infestations using one of several ways. In varroa mites, you may do a visual inspection of adult bees, uncap and examine sealed brood, uncapping worker brood, or doing a drone pull. Ether roll, powdered sugar roll, or using sticky boards for tactical mite, for tracheomites, dissection and examination of the trachea, checking for scarring. Determine your thresholds. Research has shown that 5 mites per 100 bees is about the threshold for a healthy colony. And assess your options. Using your best mite management tools, make a decision on how best to control a mite population. This is a personal management decision based on your knowledge of bees, conditions in your apiary, your understanding of the pests, the host, and their interactions. And the treatments available. It's an approach using multiple tactics. Select and apply controls. Once options have been considered, you select your course of action using chemical controls or some other method as the tool of last resort, and only when other control tactics have failed. Monitor, evaluate, record. Results. Effective controls for varroa mite control are rapidly changing. Keeping a record of your use of the various techniques you have used in your apiary will allow you to evaluate controls, the ones that work best for you. Keep a logbook. It's the most important management tool. So there's several tactics within IPM. In this case, we're talking about varroa mites. So host resistance, intertolerance, that would be a cultural thing. Requeen with resistant queens. Some kind of bee that does comb calling. Does mite pulls, uncapping. Modify environment, so drone cone removal, screen bottom boards, heat, keeping your bees in a sunny place, powdered sugar. You say powdered sugar, we'll get to that in a second. Think about your natural enemies, biological, so fungal, pathogen, or nematodes, pesticides. You have a whole variety of different kinds of pesticides, but you think about it in this order. You're thinking, are the bees resistant? Do I have the best stock for my situation? If you have that and you have an issue still, then physically modify your environment. If that doesn't work, consider a biological control before you turn to the chemical treatment. All right, so varroa mites. To a bee, they're about the size of a soccer ball. And they affect the outer casing, the exoskeleton of the bee.
We'd like to introduce you to the varroa mite. Well, to be precise, we'd like to introduce you to millions of varroa mites. Why so many? Well, when it comes to the varroa mite, there's no such thing as just one. And if you'd like to see how an infestation starts, you need to look no further than your own bees, especially drones, as they're pretty social and like to travel between hives. Since Varroa mites don't have any wings of their own, they slip into hives by hitching a ride on the backs of adult bees. And for the lucky mite, the trip includes an in-flight meal, as Varroa mites will begin feeding on bee blood within a few minutes of clinging to the bee. Once they've entered the hive, Varroa mites slip undetected into the vulnerable, uncapped brood cells. This is where the mites lay in wait until the bees cap the brood. Once a cell is capped, the mother mite, like a tiny vampire, climbs atop the cocoon of the developing bee, tears open a hole, and begins to feed on its blood. Within 3 days, the mother mite lays her first egg, which always develops into a male. Then, she lays one female egg every 30 hours over the next week or so in her newly acquired home under the brood cap. And as each of these female mites mature, they mate with their brother. By the time the baby bee develops and leaves its infested cell, as many as 3 fertilized mites will emerge with it, and the cycle continues. Using this strategy, the Varroa mite population can grow as fast as the bee population it feeds on. But when summer ends and the bee population declines, the hive is left with a huge mite population, and that's dangerous. Too many mites in a hive will overwhelm and kill entire bee colonies. So what does that mean for everyone's favorite insect, the honeybee? Well, honeybee colonies with heavy mite infestations can't effectively pollinate or produce honey because they suffer from diseases and viruses transmitted by the mites. In fact, honeybees suffer from as many as 20 different mite-induced viruses, including the devastating deformed wing virus, which prevents them from flying. We'd like to introduce you to the Varroa mite. Well, to be precise,
So, mites can be spread by migratory beekeepers, importing packages of bees, robber bees, purchased colonies, moving things between colonies, and also just by someone in your area not controlling their mite populations to the point that there's transmission at the 4-H area. So we heard it in the video, but let's go over it a little bit more slowly. So female mites enter the cell of a 5-day-old larva. Adult mites immerse themselves in the larval food. After the cell is capped, female mites move onto the pupa and begin feeding. Females feed and lay first eggs within 60 hours after the cell is capped. The first one becomes a male mite and then lays eggs at 30-hour intervals for the duration of the time that their baby bee is in there pupating. So 1 to 6 eggs are laid, passing through 4 stages: larva, protonymph, deuteronymph, and adult. Both male and female are produced. Mating takes place within the cell, and the period from egg to adult takes 6 to 7 days for female and 5 to 6 days for the male. Adult females leave the cell on emerging bee. A male and immature stages stay in the cell. Mites transfer through close contact between the bees in the hive. Adult female mites feed through the intersegmental membrane on honeybee lymph, honeybee hemolymph, So adult female varroa mites are visible to the naked eye. They're brown or reddish. So again, part of this IPM thing is knowing your host and your pest. The brood number of your host over the course of the year. The mite number in the hive over the course of the year. The bee population, it's going to be highest May, June, July. Your mite population may start lower, but it'll go higher July, August, September. And you want to intervene before the mite population gets to the point that it's over the threshold in your colony. And if you don't treat until much later than that, it can be too late. Your mite loads will be too high and your viral loads will be too high, and your bees won't survive the winter. When I'm talking about viral loads, one of the most obvious and visible is this K-wing, or deformed wing syndrome. The wings just don't develop properly. But the varroa mite acts as vectors for many viruses. But if you see more than 2 or 3 bees with this K-wing in your hive, you just have to know that your hive is in real trouble. So regular testing is the best way to get out in front of mite infestations. Testing methods include the drone pull, sticky board, sugar roll, bee cocktail, ether roll. So if you want to do just a quick spot check, it doesn't tell you very much about your thresholds. It just confirms presence or absence. You can use your uncapping fork and dig it into some of the drone brood and yank out the drone brood, and you'll see varroa mites are very easy to spot on drone brood. So this just confirms the presence. Sticky boards are a more scientific way to actually discern the number of mites per number of bees.
I'm just cleaning up the old boards and get them ready to put the new mixture on. See, the varroa mites, they fall out of the hive and they land in the Vaseline Crisco mixture and they can't get out of it. So, if you count and check them later, you will give a good idea of what your threshold level is inside your hive. If it is too high, you really have to treat, especially coming into winter. Okay, get the roller, roll it even, and then we start rolling out the boards. It doesn't have to be really that thick. You want a thin layer. It's just enough that they can't crawl off it. What you're going to do is you're going to take a sticky board and we're going to stick it underneath the hive. We're here 48 hours later. We're going to pull the sticky boards and see what the mite count is. Here's hive 2. I haven't been able to find any Varroa mite. This is incredible. Hive 1 had 2. Hive 2 doesn't seem to have any. I'm just cleaning up the old boards.
So here's another sticky board, close up. You may have noticed the grid in the video on one of his sticky boards. So this is a close-up of the grid segment. Are you able to spot and identify any varroa mites on this grid segment? I would say no matter who this is, and no matter what they've decided their thresholds are, this is probably well above the threshold. It's 12 mites in a 24-hour fall. Spring threshold is lower than 9. So the next thing we're going to do is we're going to do a sugar roll, a cup measure. A cup measure is going to collect 300, 350 bees. Use some powdered sugar and a tablespoon. And then this is our mite counting dish, so it's a basin with a white bottom. Put a little water in the bottom of that. And then we have a mason jar with a modified screen lid on it with an 8 mesh. That's hardware cloth, number 8.
We're going to get some bees in this jar from a brood frame by brushing backward and letting them fall in. The nurse bees will fall in, the others will fly away, and we're going to try to get about a cup of bees. This jar is a mason jar that I've glued a piece of hardware cloth to the lid. We're going to add powdered sugar to that in a minute. A tablespoon or so of powdered sugar, just regular powdered sugar from the grocery store. I'm going to roll them around in that. I'm going to give them about 10 minutes to shake around like that, giving them a little shake from time to time. So it's about 300 bees. If we were using an alcohol wash, we would kill the bees, but it'd be about twice as accurate. With an alcohol wash, you want to get, if you get to 7 mites, that's too many, so I'm going to guess if I'm more than half of that, I'm going to be concerned. So we're going to shake onto the water, count mites. If I get to 7, I'm going to stop counting. Really, if I got to 4 with sugar, I'm going to stop counting, and I know I'm well over that. So we're going to treat this hive for mites soon. This is a worrisome number. We're going to get some bees in this
So an alcohol wash is very similar, except for it will kill the bees. You have to be extremely careful not to get the queen in there. It's going to be, again, about 300 bees. And you're going to put them in an empty quart jar with a screen lid on it. And you're going to close the lid, and then you're going to bump the jar down so that the bees fall to the bottom. You're going to add a cup of rubbing alcohol, and you're going to roll the jar around. And then you're going to turn that back over and dump it out onto your white surface. This is easier to count than maybe being able to see them at the bottom of the jar. They actually make special plastic little jars to do this with. Very handy, I suppose. So he's being very diligent about looking for his queen. This basin that he's using is empty. And he's just using it as a place to knock the bees down into, so then he can collect the number he needs for checking them. It's kind of a weird recipe. I'm not sure I would want to digest that. He does something I think is very silly. He holds the whole alcohol over the hive to pour it. It would make more sense to hold it over the ground. And now he's just going to stir them around. Again, this is a much more accurate way to count, but it does kill the bees. And you don't need to splash alcohol on your hive. You could actually hold your alcohol away from your hive. And he's using isopropyl alcohol. So taking an ether roll is also very similar to that. You just capture 300 bees in a jar. Make sure there's no queens in there because the ether is going to kill them also. Ether is a similar stuff to starter fluid. Just use starter fluid, it's fine. Close the lid, bump the bees down, open the lid real quick, squirt in some ether. You don't even need a screen top for this. Squirt in some ether, a couple of squirts of ether, close the lid back and just roll the jar, and then count the bees on the accumulate on the inside of the jar. I mean the mites that accumulate on the inside sides of the jar. So those are some treatment methods, and we looked at powdered sugar for checking. The way the powdered sugar works is essentially it creates a slicker surface and it makes it harder for the varroa mites to grip onto the bees. So when you are shaking them in that powdered sugar and you're rolling them and they have 10 minutes to try to work that thing off of them, they're usually pretty successful in getting most of the varroa mites off. So that when you actually shake that sugar, the vast majority of the mites will be caught up in the sugar mixture and fall through the screen with the sugar. But that same process of the slickening of the bees using the sugar can be used if it's used early and often to reduce the number of mites actually adherent to bees in the colony. And you do this by taking a cup of powdered sugar and putting it in a flour sifter and sifting it all over the tops of the frames of all the boxes and then brushing it down so it goes down in between the frames of all the boxes. And this gives the bees in there a chance to get some of that slicking and sugar on them, and as soon as they can, if it helps them to dislodge the mites, they're going to be dislodging those mites and dropping them down. So if you're going to do this, it's a good idea to maybe have some chickens there to snatch up the little suckers. This is not going to be probably the only thing that you do. It may help to reduce some of your mites as long as you're not coming into the season fully loaded with mites. You may be able to use this in the first parts of the summer just to keep the numbers down. It may not be effective as your only treatment option now. I see these slides are out of order. We're about to do chemicals instead of our physical, but okay, so they're out of order. So we're talking about soft chemicals first. Soft chemicals are your thyme oil, thymol based. These are extremely concentrated thyme oils. So it's not just your essential oil that you might use to scent your house. It's very, very concentrated, and it would, if you got it on your skin and left it there, it would burn your skin. But they are used in certain products like Life Var, Apiguard, Securocide, and this formic acid is not a thymol based. It's formic acid is the same stuff that ants make as their neurotoxin, which impacts the mites and not the bees. Additional soft chemicals are the oxalic acid. Oxalic acid can't be used at all times. It has to be used when there's no brood. And you can either do a vape or a drip. You can use it on packages. You can vaporize your packages to kill phoretic mites that are on the bees in the package. Your application methods are your dribble, spray, and vapor. The vapor requires a vaporizer, which is essentially like a flat plate that gets hot. And your dribble is done with a turkey baster or a large syringe, and you essentially just dribble it over the colony. There's some harder chemicals that are also approved for the treatment of mites. Apistan, the fluvalinate, Apivar, which is amitraz, and Checkmate, which is Comaphos, which is the same thing that they use on other larger livestock and are pretty toxic to people and can be toxic to your bees. So these are the things that we should have discussed before we discussed any of those chemicals. We have our alternative treatments. So these are physical and environmental factors. So bait frames, these lime green foundation frames have a foundation that's a drone brood size. So the bees will draw it out drone brood, and then if it's a drone brood size comb, then the queen will lay an unfertilized egg in there, and then it'll all be drones. And since the drones have a longer life cycle, the mites tend to prefer to nest in with the drone brood. So if you have all your mites nesting in this drone brood frame with your drone brood, once the drone brood is capped, then you can actually remove this frame and freeze it and kill the drone brood, but you'll also kill all the mites that moved into the frame. It doesn't get everything, but it's a pretty good sort of control. You can also use this on naturally drawn drone brood. So sometimes a colony will draw out a frame that has mostly drone brood on it. That's a good frame. Mark it on the top, hold on to it, and you can use that one also for doing the drone pulls. A caveat with this is that you want to make sure that you remove that frame and freeze it, otherwise you've just created a varroa mite bomb that's going to go off as soon as all those drone brood start emerging. So another technique is queen sequestration. This is essentially to take like a screen cage and keep the queen in one little area so she can't actually lay all over the place. And what this does is it gives the hive a brood break. And when you break the brood cycle for the bees, you also break the brood cycle for the varroa mite. So there's several other ways that you can do a brood break. One is when you're requeening, let them be queenless for a short period of time. Or have a virgin queen for a short period of time. Another is mineral oil liquid. Small cell size foundation was thought that if you had a small cell size that the larva would take up so much of the cell that there wouldn't be very much room in there for all this other activity to go on. It doesn't really work quite the way that the creators of the small cell size foundation had hoped. So more and more states have given the go-ahead to this hop guard, a Hivestan, and the hop guard is like thyme oil is based in thyme oil. The hop guard is from the oils from the hop plant, just super concentrated. And then mineral oil vapor is not approved in the U.S. as a treatment, but it is a treatment that is used other places, along with acetic acid vapor. So, Varroa destructor mite is still the #1 royal problem. Number one solution is mite-resistant bees. So we kind of looked at this backwards, actually, because the first thing would have been to think about our cultural resolutions like mite-resistant bees. Then we would look at physical resolutions, and then we would look at possible chemical uses. So, question though is, will you be proactive or will you be part of the problem? Because there's those only those two choices. So, cultural tactics: interrupt the mite cycle, restrain the queen. That's a queen sequestration cage. Fall requeening. Use mite reducing queens. Hygienic. VSH, varroa sensitive hygienic Russian queens. Physical tactics to dislodge mites with smoke or powdered sugar. Smoke, I'm not so convinced works very well, but the powdered sugar is very slightly effective. And if you do it early and often, it can kind of keep things a little bit more under control. But you still need to test. And if you are surpassing your thresholds, then don't just think your powdered sugar is going to do it. Biological tactics: use host-specific natural enemies, so bacteria, viruses, funguses, and nematodes. We're going to talk about nematodes when we talk about small hive beetle. Impair pest reproductive capacity. So think about if somebody could actually, well, we have the mite behavior, so your brood attractiveness, we're actually able to use that. That's that drone brood pull, where you're actually yanking out a whole frame of mite babies and freezing them. But imagine if you could, if somebody were to breed from less virulent or weaker mites, or non-reproducing mites, and then introduce those into the general population. That would put a real kibosh on the mites moving forward. So chemical tactics: less toxic chemicals, your essential oils for mite knockdown. Apiguard, Apolifar, both contain thymol, organic acids. Mite away crick strips with the formic acid, oxalic acid. Chemical tactics, and these are like your higher, more potent things. They're good to have in the tool bag in case you aren't able to control your loads any other way. You want to make sure that you get your mite loads down. So if the other things are not working, because you never use these, they're probably still going to be effective for your bees. If you use these all the time, their effectiveness is going to diminish. So, Apistan, checkmate, and Apivar, and your antibiotics, terramycin and Tylenol. Remember, if you use antibiotic treatments for anything, they have to be, you have to get a prescription from a veterinarian. Tracheal mites is parasitic, are parasitic, and this is electromagnification microgram magnification of the trachea, and you can see the mites in there. They pierce through the inside of the tracheal tube and drink the hemolymph. They're very small. They enter through the spiracle of the bee, and they affect the trachea, and they affect the hemolymph, the circulatory. They're microscopic. You can only find them and be sure that you have discerned them if you open up a bee and examine its trachea and look for scarring or presence of mites. They have beak-like mouth parts that pierce the wall of the trachea, and they feed on the hemolymph, weakening and shortening the life of the bees. Sometimes you can tell if there's heavy loads if a lot of the bees are breathing really heavily and there hasn't been an awful lot of activity. but they're really pumping their abdomens. It's like if we have a lung disease. We breathe very heavily, but the oxygen doesn't do us a lot of good because we're not able to access it. So your viral loads, many of these are transmitted again by the other parasitic mite syndrome, PMS, and associated viral conditions. Sac brood, and then paralysis. There's a whole rack of the different kinds of bee paralysis things out there. And many of those viral diseases are going to impact the basal ganglia, the brain. Some of them are going to impact other parts of the bee developmentally, such as the K-wing or the deformed wing virus. Parasitic mite syndrome, you will see multiple viruses sort of have the same action result in the bee. So the symptoms are this punctured cappings. This picture does not have a very good show of punctured cappings. The next one does, so we'll look for that. But this one, the cappings were never laid down. But you end up with these larvae that don't look healthy and pearly and glossy. They just look sort of melty and snotty in the bottom of the cells. Deformed wings usually results in the death of a colony. Sometimes it can result in absconsion, usually in fall. All your bees will disappear. There'll be brood in there, but it won't look very good. And or if you think you're catching a very healthy late fall swarm, yay! I caught a swarm in September. It may be that it was just a bunch of bees that absconded because the nest was untenable. So here we have some of these cappings that are punctured. If you look closely, you can see little sort of chew mark holes in the cappings. The bee's wings may be deformed, and you're going to see mites. If you see mites, you know that they've deposited some manner of viral load into the colony. It's just how virulent it is and how much of an impact it's going to have. The approved treatments of PMS is, hot water bottle and 2 ibuprofen. Approved miticide plus oxytetracycline plus fumagillin. Sac brood. This is not very often seen in our area. More common maybe in the mountains of Pennsylvania. Does not usually cause severe losses, and it's usually seen early in the season in the brood rearing. Punctured cell caps, dead brood among healthy brood. Punctures and cappings over dead brood are made first, and the dead brood is removed later by the house bees. They'll clean it out. Larva lies on its back with an elevated head. Larva dies in an upright position with a raised head, not like a melted thing like we saw in the PMS, but head may turn black. And essentially like the cocoon doesn't stick to the cell, it stays wrapped around the bees. It almost looks like a little plastic baggie around the brood. You can see this one, the head turned black. The one in the tweezers, the head turned black. The ones in the cells, the head just didn't turn black. But you can see they're up and they're, very strangely holding themselves. You can also see the punctured cell capping there. They're very easy to remove. They pull out really easily. Bacteria, American foul brood, and European foul brood. So American foul brood, the first thing you'll notice is the smell of death. It's going to smell like a dead body. It's going to smell like a carcass by the side of the road. It's going to be very intensively foul. Shotgun brood pattern, meaning it's very scattered and spotty. The frames will appear kind of greasy. There'll be the odor of decay. If you look at the cappings, they'll be sunken, black, greasy, melty looking. So AFB affects young larvae. After death, the cells are capped over. Death happens after the cells are capped over, so the dying happens underneath the cell. Spores form after death. It's foodborne, so it's all over the colony, and any robber bees are going to pick it up and spread it to their colonies. And a high probability of your seeing these pupal tongues. So this sort of thing that's left is the proboscis sticking straight up. You're going to test for this. So it's a dead, black, brown, nasty smelling, slimy looking scales on the bottom of the cells. Try to pull out a bee. It's sort of like difficult to remove. Every scale in there has a whole bunch of AFB spores in it. And to test for this, you will use like a matchstick or a little piece of twig or something, and you'll stick it down in the cell and stir it around in there. And when you pull that matchstick out, if there's like a snotty, mucousy attachment from the twig or the matchstick to the cell, that is a clear indication that you're dealing with American foul brood. It's an active infestation post-death, so it keeps it keeps being active even though there's no live bees to feed on. Before the scales form, it's also active, and you can get these little pregnancy test looking tests for it. Those are helpful just as a confirmation. You probably want to get your mentor or someone who's more familiar to help you take a sample so that they can send it into the Beltsville Bee Lab. And then if you can't get your mentor or somebody to help you that day, throw the terramycin on there. Get that thing covered so that at least the infection will be knocked back just long enough. 1 or 2 days maybe. I would just close up the colony and not let any of those bees out, and I wouldn't let any other bees in. And then because terramycin doesn't kill the foul brood spores, it can help to bees to kind of treat them until long enough until the terramycin wears off. But once the treatment stops, then AFB is just going to reappear. And when that happens, your colony will still be a big vector. And this says adult bees may be salvageable. I don't believe that to be the case. I think that the U.S. law is that you pretty much have to burn the whole colony in place. But you want to make sure that you have American foul brood, that it's not European foul brood, because they're two completely different animals. So with AFB, there's no cure. You check for it with regular inspections. That's why the law in the U.S. is that you have to have movable frames, is so that you can check for foul brood. Call your mentor, call somebody else in the club that knows what they're doing. If affirmed, call the state inspector, trained veterinarian. Spores can remain virulent for up to 8 years. You can't reuse any gear. you should probably burn your gloves. The only pathogen that allows state inspector to quarantine the whole area. It's not the only one that allows them to burn the colony, but DC law stipulates that hives must be burned when AFB is confirmed by the inspector. End of. So, remember, there is no cure for AFB. Terramycin provides a holding pattern only, just long enough for somebody who knows a little bit more than you do to come and check it out and make sure that it is actually American foul brood. If it is actually American foul brood, you're going to have to burn that colony. But you should send in a sample to the bee lab before you do, so that they are able to put that on their maps. Sort of like the CDC for bees. So European foul brood is a completely different thing altogether. European foul brood has contorted larvae, but they're not just completely melted and nasty and black. The diseased brood will be in open cells, and if you check out the cappings over the other cells, they're not nasty and black and greasy looking. The larvae died before they're capped. You still have that spotty brood pattern, but you have that spotty brood pattern in a few of the things that can impact bees. It can be easy to confuse, again, with parasitic mite syndrome, which has that spotty brood pattern. But you'll see it's usually like a multicolor brood, and it's stress related. So non-uniform color, and then the breathing tubes, will become more visible. And it doesn't generally kill the population, but it can if the colony is already weak, because it does cut the population. There may be some scale, but the scale is easy to remove. And you may smell a foul brood smell, but it'll be more like, old lunch meat in the refrigerator as opposed to very dead roadkill by the side of the road. Again, this diagnostic kit can be used, and it's 98% accurate. Sometimes the best thing is just to requeen the colony or relieve the stress. The stress may be just dearth. Maybe there's a lack of food in the environment, so maybe give them a little feed on their hive. Sometimes you may have to use terramycin to knock back the fungal load, but not necessarily always. It's still worth taking a sample and sending it into the bee lab. But it's a fixable thing. EFB, European foul brood, is a fixable thing. All right, so fungal issues, chalk brood and nosema. So chalk brood looks like chalk, brood that looks like chalk. You'll often see these white chalky looking things in front of the colony. Again, you're going to have a spotty brood pattern. And this is caused mostly by damp conditions. So get the bees into a different location, less moisture, more ventilation. You may end up requeening, and you may end up replacing some of the comb, cycling out some of that comb. So nosema, there's two different kinds of nosema, and they both affect the hindgut of the bee. And you'll see this on the outside of your colony. With apis, you may also see it on the inside. Bees are so clean, they do not like to defecate in or around their colony. So if you see this, there's some kind of an issue. So feces on the inside and the outside. Stress can trigger it. Dysentery spreads nosema, so that's, it's like if we don't wash our hands, we'll spread whatever rotavirus we've got going on. The results in increased winter losses and reduced lifespan of the bees, and decreases honey production, and you wouldn't want that honey. They don't really want that honey either. Nosema cerana, adult bees are targeted. No outward warning to the beekeeper. The two spores for the different nosema are shaped differently. Cerana is more virulent than Apis, and it can kill a colony really fast. It can be triggered by stress, so it may be present in the hive for a long time and not express itself until the bees are stressed. The best defense, as with many things, is a strong, healthy colony. So for nosema treatment, you need fumagillin B, strong winter colonies. You can feed them the fumagillin B in syrup. And you can also give packages fumagillin B before you install them, just to make sure that they're good to go. So conditions: dysentery, chill brood, colony collapse disorder. Dysentery sure looks a heck of a lot like that cerana stuff, huh? I mean that, yeah, the Apis or the cerana nosema. The condition is not a disease. It's just damp, poor drainage, prolonged confinement during winter. So if it's several days of cold weather and the bees really need to go, they may have a hard time holding on to it as they're exiting the colony. And then maybe food that has slightly higher water content. And again, and this is impacting their hind gut. And then chilled brood is what it sounds like. The brood gets cold and it causes, it's not a disease, and can be caused by underfeeding of the larva. That's if the bees aren't able to reach enough food to feed themselves and their larva because they're trying to keep the larva warm. There may be food in the colony, but they're not able to get to it because they won't leave the brood. And then that just might be that there's not enough adult bees to cover the brood. So you want to minimize the work for the bees in cold weather, make sure they have plenty of food. In early spring, keep frames in the original order, so don't go in and change things around and confuse the bees about where they need to cluster to keep the brood warm. And then if you get in the habit of pulling a frame out to do your management, just don't do that in winter. Colony collapse disorder, dwindling. Could have been colony collapse disorder, no matter the season. No firm diagnosis. Major losses, especially in commercial operations, and some in sideliner, but not usually in the backyard beekeeper situation. Seems like it's triggered by stress, and it seems like it's a bunch of other things that are all kind of working together. And the bees will just disappear out of your colony. What happened? Oh, there were bees here. There's no bees here. And it almost seems like they absconded, but the best defense are, again, strong, healthy colonies. So your secondary pests: wax moth, small hive beetle, ants, cockroaches, rodents, snakes, bears, and teenagers. The wax moth is a beneficial insect in the ecosystem. It actually has the ability to digest plastic. It's really great at cleaning up feral colonies, so if there's an issue in the wax in a feral colony, they'll go through and they'll clean it out, and that relieves any disease load in the feral colonies. Also, will eat any extra wax that you leave lying around. So if you want them to eat it, okay. If you don't want them to eat it, you have to take some precautions. And they're really only a danger inside of a weak colony, and that weak colony might be a strong colony if it were in a smaller box. So just make sure you right size your hive. It could be a risk to stored brood comb. What you would want to do is pull that brood comb out, stick it in a plastic bag, and then freeze it for a few days before you store it. And that way it'll kill any of the wax moth eggs that may be there. If they do get in and they start pupating on your woodenware, the pupa can cause a little bit of damage to your woodenware. So you can use chemical controls, but the chemical controls are really nasty. They will contaminate any honey and pollen. They can contaminate your wax. So your alternative treatments are your best ones. In this case, you can see this pupal damage to the tops of these frames. That's the damage that could potentially weaken your woodenware, although mostly it just makes it a little more difficult to clean. Brood or honeycomb, freeze for 1 week, remove and thaw, then store in sealed plastic bags. So you're going to pull it out, you put it in a bag, freeze it, thaw it, stick it in a box, still in the bag, and you just keep it sealed up. Dry honey supers, you can store those in a light, airy location. My grandfather used to, instead of stacking the boxes flat on top of each other, he would stack them on ends so that the frames would rest perpendicular to the ground and be exposed to light and air. Not direct sun, but. And then screened inner covers on active hives are helpful for keeping things airy. And then strong colonies are a must. Small hive beetle never used to be as big a problem as it is today. It seems like it's become more and more of an issue lately. They lay their eggs in the cell. They don't necessarily predate on the pupa, but when they develop, they turn into these little, they look tiny, sort of like tiny black ladybugs. The larva is much smaller than the wax moth larva. But they can really slime out a hive. They really like to eat the pollen, especially, and they will predate pupa, but not usually. And there they are. There they are. There they are. So the life cycle of a small hive beetle: young adults can fly into a colony. They detect it by the odor. They hide in small spaces, crevices, cracks, and the adults lay eggs. They hatch in 2 to 4 days, and the larvae exit the hive, and they spend their pupal stage in the soil, and then they grow up to be adults that can fly back into the colony. Treatment for small hive beetle: these beautiful beetle jails or oil traps. They fit between the frames, and you put some oil in there. The bees will chase them in there, and they'll get coated in the oil and drown. They have these bee barns. They're sort of like cassette cases, CD cassette cases. I've actually seen people modify CD cases, and you essentially have a oil trap on the inside of that. The bees will chase them in there and keep them in there, and then you have to empty the cassette. I don't know about this gold star. I've never used it, but it's there. You can learn about it if you want to. And the oil trays are at the bottom, so there's something that looks a little bit like a queen excluder, but it's small enough that the bees can't get through it. But when the beetles drop down, they fall through and into that oil tray and drown in the oil. But your best defense is going to be strong colonies. Also, you can use untreated, unscented Swiffer sheets. A portion of those, the bees will chase them in there, and the way that the fibers are configured entangles and snares the hairs on the small hive beetle, and it'll keep them entrapped there. Then you can chuck the Swiffer sheet. And then diatomaceous earth used on the ground for the pupal stage. And the other thing that you can use are beneficial nematodes. There's 3 different kinds of beneficial nematodes that will eat small hive beetle larvae in the ground. The life cycle of the nematode is parasitizing a small hive beetle larva in the soil. You apply them with water onto the soil around the hive. So day 1 is the application. Day 2 and 3, those nematodes will attack the small hive beetle larva, start predating on it. Days 3 to 6, they're going to kill that larva, and then they're going to procreate and reproduce, and then they will escape the dead body into the soil. And then when another small hive beetle comes along, that larvae will be predated by the nematodes that are in the soil. So you can apply these nematodes fairly early after the thaw, and as long as you keep the soil moist, not wet, but moist enough, they will continue to live in the soil throughout the season. So they can really help a lot with your small hive beetle issues. Also chickens. Chickens are great. So some of the other predators that you might find: skunks, mice, raccoons, possum, and bears. Mice are probably the worst. They can get inside of the hive through a pretty small slit. They'll come in when the cool weather hits, and they'll do a lot of damage inside of the colony, chewing up the woodenware, eating the beeswax, and the bees won't mess with it because they don't like the way it smells. So they're very clean, but they won't clean that out. So you can use a 1/2-inch hardware cloth. If you do, you want to make sure that the bottom rung of the hardware cloth has been cut off and it's just these posts that are coming down. You can staple it to the front of your hive, and if you leave these gaps open with no rung or post on the no rung on the bottom, it'll be easier for the bees to clear out any debris that's in the hive. And you want to make sure that you don't staple the top box, the brood box, to the bottom board. It becomes less efficient to clean the bottom board if they're stapled together. Anything like a skunk or a raccoon or a possum, if they're harassing your bees, you will know that something has been there because the bees will try to light you up before you get to the entrance of your apiary. Especially normally docile bees, if you suddenly find that they're pretty hot, you may wonder if maybe a predator was sniffing around. But skunks, weasels, buzzards, raccoons, possums, they'll come and dig around under the colony and try to eat bees. And this is a skunk cud that's left on the ground here. And you can use fencing. You can elevate the hives up above where they can reach to, especially for the things like the possums and the skunks. Carpet tack strips on the front of the colony can deter critters. Chicken wire that kind of covers over the entrance and drops all the way down to the ground. Have a heart traps. You can call your local animal control, see if they can come out and trap your pest. So the debate continues. Teenagers, what's worse, teenagers versus bears? And we have 2 sets of destruction, and they actually both look pretty similar. The one on the left still has some colonies standing, though. The one on the left was done by a bear that just wanted to eat honeycomb. And the other one was done by teenagers that wanted to just do some crazy madness. It's just you have these fully developed bodies without the fully developed frontal lobe to go with them. Teenagers can do crazy things.